Hey everyone, this is going to be the lecture over genetics of multiple genes. In other words, what actually happens in your body. Humans are extremely complex as compared to peas, which are just wrinkled, smooth, green, yellow. In humans, we have thousands of different skin tones, hair colors, and so many different eye color variations that one Punnett square cannot encapsulate all of the different possibilities. So in this lecture, I'm going to give you a little bit of insight as to how we can start doing some more of the intense human genomes with two different genes acting at the same time. So make sure you've got that title slide written down. At any point in time, if you need to pause this video in order to get everything written down, YouTube should allow you to do that pretty easily. So the law of independent assortment. This means that not all of our genes are on one chromosome. So for blood typing, I have a gene for A, B, or O blood type on one chromosome, and I have the Rh gene, which means that you're either positive or negative, on another chromosome. These two things are inherited separately from each other. So I could get my A blood from my mom, but my positive from my father. Or the other way around. I could get the positive from my mother or the A blood from my father. It really just depends on what my parents have in their genotype, as well as what genes the chromosomes lie on, or what chromosomes the genes are on. Genes are on different chromosomes. They separate independently from each other during meiosis. So I have two different pictures here showing the separate probabilities. For the first one, blue being chromosomes I got from my father, red being chromosomes I got from my mother. If we only look at two chromosomes here, so this individual, we start out with 1, 2, 3, 4 chromosomes, and then we become haploid, so we start out diploid, then we become haploid only having 2 chromosomes. During the first round of meiosis and anaphase, when our cells start to separate those chromosomes, I'm going to either end up with both of the chromosomes that my mother gave me in one egg and both of the chromosomes that my father gave me in another egg, which means after the second round of meiosis, then I'm going to end up with haploid cells that once again only have my mom's material, and then another haploid cell that only has my father's material. Now, and the other possibility, they're a little bit more mixed up. During prophase 1, they combine. This is showing metaphase 1 right here, where all the homologous chromosomes are meeting in the middle. And then when they separate, I have what looks like an even split between information that my mom gave me and information that my father gave me. And then when I'm down here at the bottom and I have these gametes, or the egg cells, or the sperm cells, then I have a combination between chromosome that my mom gave me and chromosome that my dad gave me. Now, in reality, humans have 46 different chromosomes. So there is countless probabilities between how many chromosomes I'm going to get from my mother versus how many chromosomes I'm going to get from my father. I could get chromosome number 1 from my mom and chromosome 2 from my father. Or I could get, odds are very limited, all of those chromosomes from my mom's mom and all of the chromosomes from my dad's dad. So it is very, very complex, which is why you are not identical to your brothers or sisters. Or if you are close in nature, you might just look really similar because you've had a lot of those same chromosomes from each other. So independent assortment. This is kind of my way to help you guys visualize it again of all of the different possibilities. Once again, only having 4 chromosomes in this original cell. In humans, we have 46. So we would have 46 different colors. So when we go through meiosis, we end up making all of these different possibilities. Each organism is going to carry a pair of alleles. You're going to see that on your test. Each organism is going to carry a pair of alleles, and that is because we get one of the alleles from our moms, one allele from our mom, the other allele from our father. So, this is the magical dihybrid cross. Di meaning 2, hybrid meaning mixing those 2 genes together. So, here we have 2 genes. We are showing those with the R's and the Ys. The R is going to reference the round. The Ys are going to reference the color. So, round is dominant. Yellow is also dominant. Green is recessive. Wrinkled is recessive. So the way that we do this, we mix together the two separate genes. So the inheritance pattern is color and texture that we are tracking. Two separate genes. Color is one, the two alleles being green and yellow. Texture is the other, being wrinkled or round. So the genetic traits are not always blended together in subsequent or like later on generations. That's what subsequent means. But we do see them getting passed on, and sometimes they do get a mixture of both. So the way that we start this out, I'm going to have my parents. So I know that my father and mother are both heterozygous, meaning that for R, they've got a dominant and recessive, and for Ys, they have a dominant and recessive. So both of these together, dominant, recessive, dominant, recessive. This is when you can go through and FOIL. Hey, that's a math term that we're actually going to be using in here. So I want you guys to draw like pretend parentheses around these. So parentheses around the R's, parentheses around the Y's. We are making sure that we're keeping mom and dad's genes together. And then we do first, outside, inside, last. FOIL. F-O-I-L. First, outside, inside, last. Do not be tempted, and well, I know you are going to be tempted. Don't do it. Don't mix mom and dad's genes together with FOIL. Keep mom's genes together in one FOIL and then father's genes together in a completely separate FOIL. So here we've got the first and the last, or the first actually, both the R and the Y, those are the first ones. The outside, capital R, lowercase y, coming from this one. Inside, lowercase r, uppercase y, and then last, lowercase r, lowercase y. So you're going to do that for both parents, and then just like we've done in the past, you're going to drag and drop them all together. Make sure you're keeping the letters together. So, this is another one. We're going to practice now. I want you guys to give this a try, and remember, you are going to FOIL this. Just that. You can only FOIL with 4. FOIL with 4. And then you're going to put those across at the top. I would like for you in your notes to go ahead and try this. So pause the... Did you pause it? Okay, I hope you did. If you didn't, please pause it right now and actually give this a shot. Alright, so hopefully you're good. Let's check your answers. A, B, A, B, A, B, A, B. Notice we have one of each gene on here. One A gene, one B gene, one A, one B, one A, one B, one A, B, one B. And we FOILed this in order to get it across the top. So then, next thing you're going to do is you're going to FOIL these, and then you're going to put these over to the side. It's going to be the same process because it turns out both parents have the same genotypes. So, AB, 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 AB. And then you drag and fill them in. So in the end, this leads to a lot of different phenotypes. Phenotypes being the physical appearance. All of those are going to look the same because all of those have a dominant A and a dominant B. Those have dominant A's, but they have the recessive trait for B. Recessive trait for A, but the dominant for B. And then that one is completely recessive through and through. I know that these are difficult. I want you to try to summarize independent assortment, meaning that our chromosomes have different genes on them and they can get separated all on their own. Think about like assortments with Forrest Gump. It's a really good movie. I highly suggest it if you've not seen it yet. But Forrest Gump, his mom always talks about how life is like a box of chocolates. You never know what you're going to get. An assortment of chocolates, Valentine's Day is coming up, you don't know what necessarily is inside of that box of chocolates. You could have what looks like a chocolate and chocolate inside, but when you bite into it, it could be raspberry, it could be peanut butter, it could be caramel. You don't know. It's all assorted. It's all random. So that's what we see with our chromosomes. You never know what you're going to get. Our genes will separate on our chromosomes independent of one another. So then, for the second summary question, I want you to look back to your old notes about the law of segregation. The law of independent assortment and the law of segregation are so similar. Try to figure out the difference between these two. I'm not going to give you the answer here because I think that you can sort it out on your own. If it helps, just write both definitions side by side in a nice little T-chart. Good luck. You are welcome to message me on Schoology if you do have any questions. I will try to get to those as soon as possible, but I am at a leadership conference, so I may not be able to access my computer regularly throughout the day, but I will do my best to maintain contact with you guys. Please be good for the sub, and I will see you on Monday to continue practicing with Punnett squares.